Namaste, everyone, and welcome to this last session of the second week of the International Yoga Conference, hosted by the American Academy for Yoga and Meditation. For those of you who are not familiar with it, let me tell you about the American Academy of Yoga and Meditation. It's a not-for-profit organization whose main objective is to improve human well-being. And we do it by bringing together a group of professionals, both medical and from various allied professions, as well as yoga practitioners and researchers together to disseminate the knowledge about yoga and what it does for us and to improve our well-being. And today, very fittingly, we have in this session two very eminent speakers who will be telling us about how yoga actually, what it does to us on a very basic molecular level. Now, all of us know yoga is great and it helps us improve certain conditions, it improves pain, this, that, and the other. But how exactly does that work? And that has not been quite well explained, probably because people don't really pay attention or are not looking for that information. But here we are, actually getting proof of what it does to us at a very basic level. So before we do that, let me introduce so the two speakers are Dr. Rima Dada, who is a professor at the All India Institute of Medical Sciences. Namaste, Dr. Dada. She's a very eminent molecular biologist. He has established the Molecular Reproduction and Genetics Facility at All India Institute of Medical Sciences. The other speaker is Dr. K.K. Deepak, who is also a professor at the All India Institute of Medical Sciences. He is the head of the Department of Physiology. And our moderator for the day is Dr. Anil Saxena, who is Executive Director of the Cardiac Pacing and Electrophysiology Department at Fortis Escorts Heart Institute, New Delhi, where he has been working for the last 32 years. He is a renowned expert in the management of cardiac arrhythmias by catheter ablation, and he is one of the pioneers in biventricular pacing and ICD implantation in India. Dr. Saxena has served on various guidelines and consensus document writing committees, and is a regular faculty in international and national conferences. He has served as president of the Indian Heart Rhythm Society. His current positions include associate editor of PACE Journal, that is Pacing and Clinical Electrophysiology, chief editor of the Asia Pacific Heart Rhythm Society newsletter, and he's also on the editorial board of various journals, including the Journal of Interventional Cardiac Electrophysiology, Indian Pacing and Electrophysiology Journal, Indian Heart Journal, Cardiology Discovery, and Arrhythmia Academy. I'm going to hand over to Dr. Saxena, and he will be introducing each of the speakers as their turn comes. Thank you, Dr. Saxena, and namaste and welcome to everyone once again.
Thank you, Dr. Raji. It's indeed a pleasure to be with all of you this evening, and I welcome all the audience and, of course, my faculty, Dr. Deepak and Dr. Dada, to this symposium. This is a very special session on mechanistic view of yoga in terms of autonomic nervous system and genetics. Now, friends, we know that yoga is a very ancient Indian science which is rooted in our culture. And there are so many studies which have shown the beneficial effects of yoga on human mind and body. Now, let me explain in brief what do we mean by mechanistic view of yoga. Basically, some parts of yoga, they affect your spirituality or your mind, and they may be difficult to measure in terms of conventional metrics. But there are other aspects of meditation and yoga which affect the physiological processes in the body, which can be measured by conventional metrics. So I'm not an expert on yoga and meditation. I'm a cardiologist. But we understand that increasingly we are looking at the importance of autonomic nervous system as a link between the brain and the various physiological processes. So the cognitive functions of the brain and various processes, they are interlinked. And we have not paid much attention to how to handle the autonomic nervous system and the various abnormalities. And we have two very renowned experts here. Of course, Dr. Dada will talk about the genetic part of it, but Dr. Deepak, who is a very distinguished faculty from All India Institute, a renowned expert on physiology of autonomic nervous system, he will be telling us about whether yoga makes humans more tolerant to autonomic insults. As already mentioned, he is head of the Department of Physiology at All India Institute of Medical Sciences. He has done his MBBS from Bhopal and MD from All India Institute, and subsequently he was awarded DSc from Sivyasa University from Bengaluru. He has been in the Department of Physiology at All India Institute since 1987, and is also Dean of Examinations at All India Institute. He was instrumental in setting up the Autonomic Function Lab in the Department of Physiology, and started various programs like the heart rate variability and measurement of vascular tone by blood pressure variability. And his team has developed a cloud-based program for centralized HRV system for the country. He has been interested in medical research and has worked with Indian Institute of Technology and other engineering institutions to develop various simulation models and EMG biofeedback, etc., for various diagnostic and therapeutic purposes. And he also has worked on brain simulation by using vagal stimulation and yogic breathing. And currently his group is exploring the role of HRV biofeedback in various disorders. So it's a very fascinating field that he has been working in and has numerous distinctions and awards to his credit. He has published very extensively and has 187 full-length index research papers, more than 3,400 citations. So welcome, Dr. Deepak. It's indeed an honor to be with you in this session. And with this, I think I will hand over the proceedings to you. We can listen to your lecture. Thank you so much.
Does yoga make humans more tolerant to autonomic insult? This is the topic for my today's talk. Good morning, everybody. I am Dr. K.K. Deepak. I am professor at the Department of Physiology at All India Institute of Medical Sciences. We have been working in yoga for over 3 decades, and we're trying to work on physiological aspect of yoga. So I'm going to talk about how does yoga make more resilient, how does yoga make autonomic system better and better. What has been known earlier that yoga affects cardiovascular variability. Yoga, which includes asana, pranayama, and meditation, it has been shown that they make autonomic nervous system better at rest. What has been shown earlier that yoga practice enhances vagal activity, as shown here, decreases sympathetic activity, and therefore this can be recorded in terms of better heart rate variability, which is a reflection of cardiovascular autonomic tone. It also enhances baroreflex sensitivity, which is a measure of how best we react to changes in autonomic insults, namely for change in blood pressure, how do we cope? So this is reflected in baroreflex sensitivity. Further, additionally, it has been shown that practice of yoga, pranayama, meditation also decreases the stiffness of arteries, which means arteries becomes more flexible, which is better. And this is reflected in blood pressure variability recordings. So we were thinking how dynamic changes in blood pressure, which occur during performance of asanas and pranayama, make people more resilient or more reactive or less reactive to autonomic insult. Therefore, the questions we raised: do yoga practitioners have different autonomic profile at rest and during slow breathing? Do they have different autonomic responses during 70-degree head-up tilt, which is an orthostatic challenge? Do they have better adaptability to -40-degree lower body negative pressure, by which we shift the blood from upper part of the body to the lower part, which has been used in microgravity? So with these three questions. We had a specific three hypotheses, and therefore we started working on this. The study which we carried out here last couple of years was aimed to decipher the role of yoga on cardiovascular variability during slow breathing, that is 6 breaths per minute or 0.1 hertz, in yoga practitioners at rest during orthostatic challenge and lower body negative pressure maneuver. The place of study is Autonomic Function Lab, Department of Physiology, All India Institute of Medical Sciences, where I work. The candidate who carried out this study is Dr. Ansuya, who pursued PhD in this field. It was funded by Ministry of Ayush, Government of India. It has ethical approval from AIIMS and also was registered under CTRI ICMR India. The further details are as follows. It was a cross-sectional study. The outcome variables were heart rate variability, blood pressure variability, baroreflex sensitivity during spontaneous and slow breathing. Study groups included naive to yoga group who have not performed yoga by certain criteria which we have laid down, and yoga practitioners, those who've been performing yoga with laid down criteria which was well defined. The age groups were similar in the age of 31 and 29 mean ages in the two groups respectively. The protocol was slow breathing protocol at the rate of 0.1 hertz, and maneuvers used to study these two stages of breathing and slow breathing and normal breathing was supine rest, 70-degree head up tilt for 5 minutes, and minus 40-degree millimeter of mercury LPNP for the same duration. The methods which we used well established inclusion and exclusion criteria. Most of this work has already been published. We used healthy subjects, age group between 20 and 50, or doing yoga practitioners and naive to yoga individual, as I said before. And we excluded criteria those who had any autonomic problem. The selection criteria for trained yoga practitioners was very robust, and we made sure that those who were included doing yoga practice. So the criteria was they have been doing yoga for 3 months continuously with a regular practice of asana and pranayama every day, minimum 3 days in a week for more than or equal to 20 minutes duration. Ability to perform certain physical maneuvers as listed here in terms of asanas, and ability to perform breathing practices were tested before they were included in yoga practitioners. The results were described in 3 sections. The first one is autonomic profile at rest and during slow breathing, autonomic responses during 70-degree head up tilt, adaptability to minus 40 lower body negative pressure. So the first one is at supine, the respiratory peaks during slow breathing were recorded. The upper panel shows naive to yoga group, that is A, and yoga practitioners, that is B. What you see here is in yoga practitioners, this is equally distributed over the wide range of respiratory frequencies, starting from up to 0.35 or 0.4 hertz, which was not the case with naive to yoga group. So they were better dispersed. And when the challenge was given by slow breathing, the peaks in both the groups shifted to 0.1 hertz as expected. So what did we find with the slow breathing? We found that during slow breathing, in a yoga group, they have shown lower heart rate, lower mean blood pressure, and lower standard deviation of successive difference in systolic blood pressure, which means they were not showing much blood pressure fluctuations, and they had a lower heart rate and blood pressure. So that's a good thing, what anybody can expect. Also, we recorded that they shown higher levels of parasympathetic tone, which is reflected in PNN 50, which is percentage of normal to normal, 50% of intervals counting, and also the baroreflex sensitivity recorded by both methods, sequence method and spectral methods, were higher in these group, and they also had higher intervals, which means bradycardia. At supine rest, baroreflex sensitivity in yoga practitioners is higher, as I told before, and it has been recorded both in spontaneous breathing and during slow breathing. So an immediate augmentation of parasympathetic activity and baroreflex sensitivity was observed with slow breathing in both the groups. Slow breathing in yoga practitioners has a synergistic effect on parasympathetic activity. This work has been accepted for Indian Journal of Medical Research. The second was to look at what happens to these yoga practitioners when we give a challenge, orthostatic challenge in terms of 70-degree head-up tilt. What we find here that the yoga practitioners subject had higher RR interval for the given test situation, which means a level of bradycardia, higher level of parasympathetic tone as reported by RMSSD, SD1, and spectral activity or spectral and sequence method baroreflex sensitivity was also higher. And we also recorded the delta changes in RR interval and BRS, which is shown here. So baroreflex sensitivity in yoga practitioners was better. The delta change in RR interval, which is shown here, it clearly shows that the delta changes in RR interval was higher in yoga group, which means they were operating on heart rate instead of operating other, which is a parasympathetically controlled. So delta change in RR interval and BRS suggests that amount of parasympathetic withdrawal to be higher in yoga group during sympathoexcitation. Head-up tilt is a sympathoexcitation situation, lying to standing or head-up tilt. Therefore, they operate on parasympathetic range. The delta change decrease in parasympathetic activity and BRS was higher with relatively stable blood pressure, predicting that an adaptive response to orthostatic challenge by yoga practitioners. The third component was to see how do they behave when the lower body negative pressure is operated upon them. So LBNP is a simple device in which half of the body, lower half, is put inside the machine and the machine produces a negative pressure. This is a commonly used device as a counter measure for microgravity research in a space physiology lab. So we perform this maneuver. This is standard maneuver for countermeasure for microgravity insults in which blood shifts to headward. This shifts blood to the lower side. So what did we find? We find that the heart rate during supine and LBNP was lower in yoga group, and also blood pressure fluctuations, which normally seen in anybody, were lower in yoga group, which means they were more stable and it did not show much heart rate changes. And parasympathetic activity as recorded by PNN50 during supine rest and LBNP was higher in yoga group. Also, the baroreflex measures were higher in yoga group. So the baroreflex measures were recorded by using standard criteria, and also sequences were counted. Here, what we're showing the downward sequences, which means the down sequence of baroreflex were identified from consecutive beats with a decreased systolic blood pressure with a corresponding decrease in RR interval. Therefore, they are called down sequences. There was a delta increase in number of down sequences in both the groups, as shown here in the graph. The increase was higher in naive. The increase was higher in naive group compared to the yoga group, which means yoga group was doing better even here. So they showed better adaptability to LBNP by yoga practitioners compared to the yoga naive groups. The yoga module might be helpful in condition of hypovolemia in healthy subjects. Thus, prior yoga practice would be beneficial in clinical conditions associated with sympathetic dominance, wherever this happens in diseases, impaired baroreflex sensitivity, this happens in diabetes and other situations, and also orthostatic intolerance, which happens because of the drug induced, or it could be because of disease, like hypertension who are undergoing the medical treatment, or diabetes mellitus. So in conclusion, we conclude yoga practice provide individual with better autonomic profile at rest, and they are better adapted to challenges imposed on autonomic nervous system. The implications of this study are large. During autonomic insult at Earth, it happens at several point of time, like the state of anxiety or anger, or even doing extremes of exercise. This happens normally, and which is a challenge. During autonomic insult in adverse environment, like exposure to warm environment or high altitude or several other extreme environment normally encountered by the military and other people, even sports people, and also during autonomic insult in space flight. Even space flight also produces autonomic insult in terms of shifting of blood volumes from both the side to head side or the lower side, depending on the seat, and also the microgravity or the gravitational exposures. And the pilots they face when they go experience G, a different G, positive G or negative G. So the available scientific evidence suggests that the practice of slow breathing makes us better adapted to the situation of hemodynamic challenges and crisis. Therefore, our study is useful for both in healthy people and also for the patient or extreme conditions. So with that, I end my talk. Thank you very much for patiently listening.
Sorry, Dr. Saxena, your mic is muted.
Yeah, sorry. Thank you, Dr. Deepak. That was an excellent presentation. And as physician and as cardiologists, we keep dealing with autonomic disorders for a long time. And there's so many implications, as I can explain to the audience, as to how autonomic nervous system plays a very important link between the brain and the various physiological processes, including cardiovascular. What Dr. Deepak explained was like the challenging situation of a negative pressure or the body tilt at 70 degree. We see it all the time that people sometimes people faint in the upright posture, and that is because of a weakness in the autonomic nervous system. When we are in the upright posture, the blood will have a tendency to go into the lower part of the body because of gravitational pull. But it is the autonomic nervous system that maintains blood supply to the brain by keeping the blood pressure in the normal range. So it plays a very delicate role in maintaining blood pressure and heart rate in a very fine rhythm and balance all the time. And we see that when patients have stressful periods for a long time, various parameters that he was talking about, like the heart rate variability and all the baroreflex sensitivity and all, they get impaired. And it's very refreshing and very reassuring to see the results that he obtained from his study, that yoga practices can restore that balance. And a lot of patients who have disturbance in the autonomic nervous system can definitely benefit from it. So let me come back to Dr. Deepak. Dr. Deepak, so what are the typical situations, the clinical situations where you can see the application of these yoga practices? Of course, as a cardiologist, I see huge potential in cardiology. But where else, like in the respiratory system or the GI system or the nervous system disorders, where all do you see the potential of these practices helping with the help of correction in the autonomic nervous system?
Thank you, Dr. Saxena. Very important and pertinent question. All situations where you anticipate a partial drop in blood pressure, that is a impending partial hypotension. And this situation could be very well thought of in before. For example, you're putting a patient on antihypertensive drug, and chances that patient will be overtreated or may get overdose or may have some chances that he might develop partial drop. So that is the first thing where you can think of that, okay, this is the time he has to be advised that his ANS should be now. Something similar might happen in the history of when you are treating a diabetic patient with a cardiovascular morbidity or even multisystem atrophy, and the patient might land up in partial drop. So where the baroreflex system goes haywire, they are not able to compromise a fall in blood pressure. Sometimes even a Parkinson's, they might lead to partial drop. So, well, there are various situations where you can get it. Therefore, intervention in terms of training the autonomic nervous system is important. Although training autonomic nervous system is a long-term process, one has to you think before, or even when you have chances that patient are getting this thing, something can also be advised. Like we remember the last case, it was referred to us for postural drop, a frequent, and that was consequent to the radiotherapy, and he was referred to us from the cardiologist. And then we tested that, yeah, postural drop is much more. Therefore, during the process itself, we gave a feedback that we see your postural drop, can you do something before that? And then we advised some physical maneuvers like leg compression or cross leg, and then pressure was restored. So these are the situations where you are impending postural drop because of chronic disease or because of intervention. And so this is what where it can be handled.
Yeah, we see a lot of patients with autonomic disturbance who are having arrhythmias, both tachyarrhythmias and bradyarrhythmias. And sometimes we do very crude methods like sympathetic. We of course we give beta blockers to counter the autonomic disturbance, but we also sometimes do sympathectomies. So maybe those can be totally avoided if we have some studies by doing yoga practices that we can modulate the autonomic nervous system non-invasively. Then many of these things can probably be avoided. Do we have any questions from the audience in the chat box? I don't.
Dr. Saxena, can I intervene here?
Yeah, please. Yeah, please.
It's again a question of arrhythmia. One of friend, colleague Dr. Gautam Sharma has been working on it.
Yes, I know he's a very personal friend of mine. Yeah.
How arrhythmia occurs, that is because of higher sympathetic excitation. So the autonomic nervous system does not only improve your parasympathetic nervous system and balance, but also calms down the sympathetic excitability, which is the cause of arrhythmia or silent arrhythmia or some other thing. Like patient is very anxious, or patient have a hypersympathetic excitability, and that's what you're actually doing it. So therefore, as a chronic sympathetic denervation, if I put it in the quotes, the yoga and meditation can actually do a wonder play in those long-term interventions. Thank you.
Thank you. Thank you. I think we are running out of time, and I can move to the next part of the next session, which is another talk on yoga and effect on sperm genome and epigenome. And Dr. Rima will be delivering that talk. She is another distinguished faculty from All India Institute. She's a professor at All India Institute of Medical Sciences, where she established the molecular reproduction and genetics facility. She's actively engaged in teaching, research, and genetic diagnostic services in the institute. And she's a fellow of National Academy of Medical Sciences, International Medical Sciences Academy, Academy of Standards and Rating, Indian Academy of Biomedical Sciences. So very distinguished accolades she has received. She has been pursuing research on the genetic aspects of the infertility, reproductive failure, recurrent ART failure, and idiopathic recurrent spontaneous abortion and childhood cancers. Last 8 years, she's working on impact of yoga on genome and epigenome. So that's another fascinating and very interesting subject. She has received several national and international awards. The ICMR National Award for Outstanding Contribution in the field of genetics of infertility. She has received a Young Scientist Award at Second European American course in the clinical genetics in Croatia. GP Talwar Gold Medal and Best Paper Award in the Indian Society of Research in the Reproduction meeting, and Best Paper Award at the Annual Conference of Indian Society of Andrology 3 times. She has delivered several orations and is a regular guest faculty in many national and international conferences. Her research on genetics of glaucoma is among the top 2% in the world. So she's a very, very distinguished genetist we have with us today. She has published extensively and has 166 original indexed articles of high impact journals, 65 chapters in various books, and she has conducted a lot of workshops and has over 190 abstracts in American, European, and international meetings and journals. She's a member of various professional societies and ad hoc reviewer for various national and international index scientific journals. Welcome, Dr. Rima, to this symposium, and we look forward to listening from your very, very interesting talk. Thank you.
Good morning, everybody. Thank you for Dr. Indranil for inviting me to deliver this talk where I share my work about the impact of yoga on male infertility and how it impacts the sperm genome and epigenome and how it's so critical to understand the impact of yoga on the germ cell, especially as it is transmitted to the next generation. So now I'll begin my talk and show you what is the roadmap for today's talk. So I would be talking and explaining to you that how in the recent years there has been a very rapid decline in the male reproductive health, and how various environmental and lifestyle factors have resulted in this decline in the reproductive health, how genetic factors are linked to these factors, how the sperm is the cell in the body which is the most vulnerable to these environmental factors and oxidative stress. What is seminal oxidative stress? Why is the sperm so vulnerable? How oxidative stress causes nuclear and mitochondrial DNA damage, how it causes dysregulation in the sperm transcripts, affecting the epigenome adversely, and then impacting the telomeres, which are the guardians of our genome and are also the biological clocks, which can result in shorter telomeres, and how infertility is a psychosomatic disease and thus needs to be targeted by a mind-body intervention like yoga, and how yoga can serve as a useful adjunct in the management of male infertility, especially unexplained male factor infertility. So in the recent years, there has been a very rapid decline in the male reproductive health. This has been seen by a decline in the sperm count, in the sperm quality. Not only is the number of sperm with normal morphology and good motility decline, but also in the recent years, there has been a very rapid decline in the semen quality, which is also seen by an increase in the incidence of genital urinary abnormalities. But in addition to that, we also find that the number of men in the reproductive age group, that is about 30 to 44 years, that age group, we find there is a number of men have higher incidence of having genital urinary cancers. Now, this rapid decline in the male reproductive health cannot be explained just by genetics alone. And it's now known that there are a huge number of factors like the environmental pollutants, which are released into the water, air, and there are a number of contaminants in our food, which are actually mimicking hormones, and these are known as endocrine disrupting chemicals. Now, these chemicals are not, majority of these pollutants and chemicals are not mutagens. They actually act on the sperm epigenome and affect the expression level of various genes. And these endocrine disrupting chemicals, they mimic estrogens, and they are anti-androgenic. And therefore, when there is a male fetus growing in the mother's womb, it impairs both steroidogenesis and they later affects gametogenesis. And as a whole, it also causes oxidative stress. And in addition to these endocrine disrupting chemicals, which are not only found in the environment, we find that a huge number of products that we use daily, for example, the water that we drink from plastic bottles, the milk that we get in these plastic pouches, a number of packaged foods, these are all coated with the plastic softeners, which are also endocrine disrupting chemicals. In addition to various lifestyle factors like smoking, excessive use of cell phone, which exposes us to electromagnetic radiation, a sedentary lifestyle, high intake of non-veg food, because in the animal's protein, we have a lot of these endocrine disrupting chemicals which are built into it, because the animals graze on grounds which are having these pesticides and insecticides, which are also endocrine disrupting chemicals. And in addition to that, we find that we are increasingly having foods which are rich in trans fats, salts, and sugars, which are leading to a large population having food which is nutritionally depleted and calorie dense. And this diet is a highly inflammatory diet and exposes us to high levels of free radicals. In addition to that, we find that even younger people and the younger boys are also started smoking, and this exposes us to a huge number of mutagens and carcinogens. And in addition to that, we find that in smoke, there is, when people who smoke, they have higher levels of cadmium, which inhibits the DNA repair mechanism, which is active in sperm. Now, I'll just talk to you about the sperm and highlight that how the sperm is transcriptionally and translationally inert, and just two genes for the DNA repair mechanism are active, and even one of them has lower levels in people who smoke. In addition to that, we find that a large population is has high levels of stress, anxiety, and depression, and it actually affects 36% of our population. And this results in higher levels of cortisol, and which bears an inverse relation to various factors, which are the neurotrophic factors, which help to promote neuroplasticity. And we find that people who have higher levels of cortisol have lower levels of various neurotrophic factors like brain-derived neurotrophic factors, DHEA, and it also disrupts the circadian rhythm. In addition to that, there is an inverse correlation with the antioxidant levels, and therefore people who have psychological stress, anxiety, and depression also experience oxidative stress. So we find that in these group of individuals, you have people having either no sperm or a low sperm count like oligospermia, a low sperm count with morphological abnormalities and impaired motility like oligosteatoteratospermia. And then you do have a group of individuals having normal sperm count, normal motility, normal morphology. That is, these are the cases which are normal spermic. In these cases, they have unexplained male factor infertility. Now, I would like to explain to you why is the sperm most vulnerable to our lifestyle and environmental factors. Now, of all the cells in the body, the somatic cells and even the germ cell, the ova, they are having a very robust antioxidant mechanisms and their DNA repair mechanisms are active. However, during the development of the sperm, during spermiogenesis, the sperm sheds majority of its cytoplasm. So it loses majority of its antioxidants. So it has minimal antioxidants in its cytoplasm. And therefore, it is having a very minimal amount of defense mechanism against the oxidative stress. In addition to that, the sperm nuclear size is 1/6 the size of any other somatic cell nucleus. And during its formation, the sperm nucleus undergoes compaction first with transition proteins and then with protamines. So it has a very tight toroid this shape. And you find that because of this, the sperm nucleus has two compartments. The inner compartment is the one which is actually bound by the protamines, which is a crystalline toroid and which is totally transcriptionally and translationally inert. And then it has a peripheral portion which is bound to the histones. And this is the portion which bears the telomeres, and it also has the promoters of various genes which are critical for early embryonic development. So because of this, because the sperm DNA is transcriptionally and translationally inert, we find that majority of the DNA repair mechanisms are silent in the sperm DNA, and therefore it depends upon the oocyte to work. It works in concert with the oocyte to repair its DNA damage. In addition to that, we also find that these genes, the telomeric DNA and the genes which are having the promoters of these genes which are required for early embryonic development, they are also found in the periphery of the sperm DNA. And these two, the telomeric DNA and these genes, their promoters, are highly vulnerable to environmental insults and oxidative stress. And also because the sperm is very high in polyunsaturated fatty acids, it is one of the favored sites of oxidative stress, which actually targets all the biomolecules. So in our study, we found that there are a huge number of factors. There could be endogenous factors or exogenous factors, like I just talked of the lifestyle factors, sedentary lifestyle, psychological stress, the standard American diet, which is a highly inflammatory diet, and more people are now having that because it's very convenient and tasty. You find more and more, especially the younger population, are going in for having these foods which are so calorie dense and this is a highly inflammatory diet. Excessive use of cell phone exposes us to electromagnetic radiation, exposure to air and environmental pollutants, and psychological stress, as well as advanced paternal age, all leads to oxidative stress. And this oxidative stress damages both the nuclear DNA as well as the mitochondrial DNA. And mitochondria are the site where maximum amount of free radicals are produced. So the damage to the mitochondrial DNA is much more and more sustained as compared to that in the nuclear DNA. So now one can question that sperm does not transfer the mitochondrial DNA to the nuclear DNA. So why do we need to really talk about the mitochondrial DNA? But it's important to know that mitochondrial DNA, because it is lying in close proximity to the site of free radical production, which are produced as byproducts of the oxidative phosphorylation, the mitochondrial DNA, once it's damaged, it develops certain deletions and mutations, and then it actually produces lower ATP levels, lower amount of energy, but it produces more free radicals. And because of this, it tends to damage the lipids, the proteins, the DNA, the RNA, the centrioles, and the telomeric DNA. Now, because of telomeric DNA, the sperms actually develop shorter telomeres, and if these such sperm are used for fertilization, it can impair the post-embryo fertilization develop, and it can impair cleavage and then can affect the blastomere morphology. In addition to that, oxidative damage actually results in very high levels of this oxidative DNA adduct, which is known as 8-hydroxy 2-deoxyguanosine. Now, this is a highly mutagenic base, and you actually find that the presence of this base actually results in mutations as well as causing epimutations. In addition to that, you also find that the presence of this base causes, because of the presence of this base, which is found on the guanine, because it is found on the guanine base, it causes the presence of oxidative stress actually causes global hypomethylation. And this results in genomic hypermutability because it causes unmasking of the repetitive elements. And because of oxidative stress and the presence of this base, it actually results in higher amount of de novo germline mutations. And 75% of de novo germline mutations actually occur in the sperm DNA, and which is the main cause of a lot of abnormalities which are found in the developing embryo and in the next generation. So it is seen not only does it result in male infertility, but if such sperms are used, it could also result in recurrent spontaneous abortions, recurrent IVF failure, but also congenital malformations. It could also result in childhood cancers, complex neuropsychiatric disorders like autism, schizophrenia, and also a number of dominant genetic diseases. So now I would be talking to you and giving you some of the findings of our study, especially how in these disorders like in oligoasthenospermia and oligospermia, you have cases which are having genetic abnormalities, but in unexplained infertility, the cause is still not known, and these are the cases of normospermia. So cases with oligo and azoospermia usually have deletions of these regions, like the azoospermia factor region in the long arm of the Y chromosome. And I'll show you today during the course of this talk that oxidative stress is also one of the underlying mechanisms which also results in deletions as well as complex chromosomal translocations, and is also the cause of oligospermia and azoospermia. And our studies have shown that oxidative stress actually in the sperm DNA, you have much larger deletions which are indicative of severe spermatogenic failure, and which results in oligo and azoospermia. So in cases which are opting for assisted conception, it's important that we do vacuum microdeletions in cases with the oligospermic from the sperm DNA. So to understand what is the cause of oxidative stress, we did analyze the mitochondrial DNA, and we find that they have a higher number of variations as compared to the control cases. They also have higher levels of free radicals, the neat and the washed free radicals, as compared to the controls, and a lower total antioxidant capacity and higher level of DNA damage. And as I told you that mitochondrial DNA mutations and large deletions actually result in lower ATP production, which actually manifest as you can see that sperm morphological abnormalities. Here you can see that the sperm axonemal apparatus is partially formed, totally disorganized. In some places it's not formed. And this actually results in motility defects in the sperm. So all these factors actually have caused a condition known as oxidative stress, and I just explained to you why the sperm is most vulnerable to oxidative stress. And as I told you, the oxidative stress targets the lipids, and because the sperm is very rich in polyunsaturated fatty acids, it's very vulnerable to oxidative attack. And it also damages the DNA, and I told you it affects both the genome by causing de novo germline mutations because of the accumulation of 8-hydroxy 2-deoxyguanosine, and the presence of this base also causes epigenetic alterations. So in our studies, we've seen that there are higher free radical levels in these group of infertile men, whether it could be primary, secondary infertility, or in cases of idiopathic recurrent spontaneous abortions or congenital malformations, where there's an underlying male factor, and in all these cases, the female factor has been totally ruled out, and they are perfectly normal based on all clinical and hormonal investigations. So what we did was, we had this double-blind randomized placebo-controlled trial where we tried the effect of antioxidants. And there are several controversies regarding the effect of antioxidant intake and the impact it has on the sperm DNA. Now, what we find in this study is that when you take antioxidants, there is an improvement in the sperm motility because it affects the sperm membrane permeability and its motility. But though it affects the motility, but it is really having no effect on the DNA fragmentation index. You can see here that the DNA fragmentation index does not show any improvement. So when you have antioxidants at therapeutic doses, very, very few antioxidants are actually able to improve the DNA quality, though the sperm motility shows an improvement. Now, this is very, very important because at therapeutic doses, majority of the antioxidants are improving motility. So the sperm is able to fertilize the ova because it is able to reach the site of fertilization if it had impaired motility, but it is not able to, but it is not having good DNA quality. And though there are studies by now by Dr. John Atkin, and he has shown that there are few antioxidants which can reach the nuclear DNA and improve its quality. What's important is that indiscriminate use of antioxidants is not beneficial, and people keep on taking various antioxidants because this actually impairs various redox sensitive reactions like acrosome reaction and hyperactivation of the sperm. So we quantified the levels of DNA damage by two techniques. One is by comet assay and also by sperm chromatin structure assay using acridine orange dye. Now you can see here, now when you look at these sperm, and these sperms have long tail, it shows that there is a lot of fragmented DNA, and when there is a sperm with an intact, good quality DNA, you find that majority of the DNA is lying in the region of the head. So now when you look at the infertile cases, you can see that majority of the DNA, the fluorescence is seen in the region of the tail, showing high levels of DNA damage compared to the sperm in the fertile controls. We can also see here, these are the flow cytometry pictures done by sperm chromatin structure assay using acridine orange as a dye. Here each dot represents the sperm, and the number of sperm towards the x-axis indicate the sperm with the damaged DNA, and the ones towards the y-axis indicate the sperm with the good quality DNA. So to understand what is the cause of the DNA damage, we actually quantified the level of various proteins which help in sperm compaction, and we find that in a few cases there could be mutations in genes which are coding for transition proteins and protamines. But the chief cause of DNA damage is oxidative stress. So we were able to establish the cutoff levels beyond which a couple is not able to conceive spontaneously, and if that is about 30, and if the levels are lower, between 21 to 30, we find that the couple is able to conceive spontaneously, but then they have higher incidence of recurrent spontaneous abortions. So we quantified the level to understand the cause of the DNA damage. We quantified the levels of this base, which I told you is the mutagenic base, and it actually correlates with various unhealthy social habits like smoking, excessive alcohol consumption, and exposure to endocrine disrupting chemicals. And studies have shown beyond doubt that the presence of this base induces both mutations and epimutations. So in our studies, we actually found that now what happens that there were several studies which were done, and it showed that children who have cancer, and especially these are sporadic cancers, the cancers which are not running in the families, it was seen that in their fathers, the sperm quality was of very poor, and these were fathers who usually smoked or had high levels of alcohol. So what we did was we actually picked up children with sporadic cancers and studied the sperm DNA quality of their fathers. These are cases with sporadic cancers. The cancers are not running in the mother's or the father's family. And we actually found that these men had much higher levels of 8-hydroxy-2-deoxyguanosine in the sperm DNA as compared to men who had healthy children with no cancer. Right. In addition to that, we also quantified the level of this base in children who had retinoblastoma versus those who did not have retinoblastoma. And again, you can see that the levels of this base was significantly higher in the RV patients as compared to the controls. Now, what is this showing you? It actually shows you that when father has an unhealthy lifestyle, the sperm accumulates damage, and because the sperm DNA repair mechanism is inert, it is actually transmitting this DNA at the time of fertilization. And if the DNA damage is too much, or if the oocyte is also aged and its repair mechanism is also aberrant, it is not able to remove this base, and this base then passes to the embryo, and you find that in each cell of the embryo you have high level of this base, which results in mutations, and therefore these children are prone to various cancers and other complex, autosomal dominant disorders, as well as complex neuropsychiatric disorders. So you can see that there are de novo germline mutations and epimutations because of high levels of this base in the father, and the presence of high levels of this base in children results in post-zygotic mutations. In addition to that, when you have oxidative stress, I told you that free radicals actually target this peripheral portion of the sperm DNA, which is bound to the histones, which is loosely bound. And this is the place where you have the telomeric DNA, which is a guanine-rich DNA. And since guanine has the lowest oxidation potential, it is a target of free radicals, and therefore we find that oxidative stress results in accelerated telomere shortening. But we also found that this oxidative stress should be optimal levels, which are actually helping to maintain. So very low levels or very high levels are detrimental, but certain optimal levels are required for the maintenance of the telomeres. And in addition to that, we also found that in this infertile population, the level of the two repair enzymes, one which helps to detect the DNA damage and the one which helps to remove the oxidative DNA adducts, are significantly lower than in the controls. So we also did a microarray analysis, and we found that in these infertile men which were experiencing recurrent ART failure, we found that a large number of sperm transcripts were dysregulated. And I'll just show you what happens when these group of men underwent the yoga intervention. Now, these group of infertile men, they underwent yoga intervention. Post yoga intervention, we studied all the parameters which I have talked about right now. So we studied their normal sperm parameters, the standard semen parameters, the seminal oxidative stress levels, the sperm DNA damage. We isolated the DNA, quantified the level of the base 8-hydroxy 2-deoxyguanosine, studied the telomere length, what happens to the total antioxidant capacity, and also studied the we also did a microarray analysis, and we also did study the sperm epigenome. So we can see that post practice of yoga, there's a significant decline in the levels of free radicals. In addition to that, you can also see there's a significant decline in the level of oxidative DNA damage. You can see there's a decrease in the levels of 8-hydroxy 2-deoxyguanosine. The stress levels, the cortisol also goes down. The telomerase activity shows a significant upregulation post practice of yoga. I would like to highlight here, when telomeres shorten, there are two causes. It's a complex trait, the maintenance of telomeres. But the main shortening of telomeres occurs because of their decline and because of oxidative stress and because of low telomerase activity. And here you can see that the telomerase activity goes up and there's a decline in the oxidative stress, which actually causes the targets the telomeres. We also see the levels of the inflammatory cytokines. We find that the levels of the inflammatory cytokines go down and the levels of feel-good hormones like beta endorphins go up. We also studied the levels of the total antioxidant capacity, and you can see that post-practice of yoga, there's a significant increase in the levels of the total the antioxidant capacity. So this is very, very important. You can see that in this group of men who are given exogenous antioxidants and they really do not target the DNA, you can see here that the antioxidant capacity itself, the expression of genes which are coding for these antioxidants, is going up, and you can see that the total antioxidant capacity goes up. In addition to that, these sirtuins, which are histone deacetylases, these are considered to be longevity genes. And this was the first study to show that yoga actually causes an increase in the expression levels of the sirtuins, and which are helping to upregulate our DNA defense mechanisms and also delay testicular aging. Now, this is the first study to show you that yoga, independent of caloric intake, caloric restriction, or intake of red wine, can increase the levels of sirtuins. In addition to that, as I told you that we studied the levels of the sperm transcript, we also found that the transcripts, you can see here that various, we have just shown this, these are showing you real-time data, showing you that how they are altered in blue pre-yoga, and how these are the green, and it shows the levels in controls, and how the levels are coming down to the control levels post-practice of yoga. And here I've just shown you fold change, that how the level of the DNA, this PARP gene, whose expression level has gone up, which helps to detect the DNA damage. And one of the key enzymes of the 1-carbon metabolism also shows an upregulation. I also wanted to show you that post-practice of yoga, there has been a significant decline in the level of this base, which causes both mutations and epimutations. So this is a very, very significant finding, which shows that by practice of yoga, you can improve the DNA quality, which is not only going to decrease the number of de novo germline mutations, but will also target the epigenome. And because 8-OHDG levels, which are very high, actually cause higher levels of, it causes lower levels of global, there is hypomethylation. And when the levels normalize, then the methylation marks are established. You can see here, post-practice of yoga, we find that the levels of this methylation levels, they increase because they are re-established, and the level of hydroxymethylcytosine actually goes down, which occurs due to oxidation of the methyl base. So this helps to re-establish the methylation marks. We also did this study and studied the sperm methylome. Now, this sperm methylome studies, which we have done by next generation sequencing, actually show you that there are over 400 sites where we found that there was differential methylation, and we find that there were about 170 sites which were hypermethylated, and there were about 270 sites which were hypomethylated. And the sites which are hypomethylated were the genes which were actually involved in fertilization, embryo development, which are very critical for early embryonic events. So you can see that yoga is targeting both the genome and the epigenome, and also helping in the maintenance of the telomeres. So how do these changes take place? As I told you, one of the key factors which is actually impacting the genome and the epigenome is oxidative stress. And our free radicals are mainly produced as byproducts of this oxidative phosphorylation, which takes place in the mitochondrial DNA. So we also wanted to study the impact of yoga on the mitochondrial DNA. So we checked, we took this group of men who had autoimmune disease and also who were infertile. And also we studied different categories of individuals with various complex diseases and studied the effect of yoga on the mitochondrial integrity. And just last month, the study came out where we actually showed that how yoga can actually improve the mitochondrial integrity by upregulating the expression of various genes which help to maintain the mitochondrial integrity. In addition to that, there is mitochondrial biogenesis, which helps to improve the mitochondrial copy number post practice of yoga. And this was a randomized controlled trial. And in addition to that, there's increase in the level of nicotinamide adenine dinucleotide, which is a molecule which is very critical because it is involved in some maintenance of circadian rhythm, epigenetic regulation, and it helps to maintain the crosstalk between the mitochondrial and the nuclear DNA. And then this is the first study to show that yoga actually increases the levels of the NAD levels. We also see that there is an improvement in the mitochondrial membrane potential. And so our studies have actually shown that yoga improves the mitochondrial health. And we also find that the levels of various mitochondrial genes which are helping in the ATP production like cytochrome oxidase also increases. So we find that yoga actually improves mitochondrial health by increasing the mitochondrial copy number, increasing the level of cytochrome oxidase, decreasing, and because the mitochondria are healthier, they are producing less amount of free radicals. It increases the NAD levels, which helps to improve the crosstalk between the mitochondrial and nuclear DNA. It helps to regulate the gene expression. It is very, very important for calcium signaling and also maintenance of circadian rhythm, increases the mitochondrial potential, and improves the mitochondrial integrity. So all these factors, because there is an improvement in the mitochondrial health, which are producing fewer free radicals, fewer amount of mutagenic bases, and regulating the epigenome, we actually saw that when we followed up these men with poor DNA quality in this sperm DNA over a period of 6 months, you can see that there is a significant improvement in the DNA quality. Now you can see here that, as I told you, the number of sperm towards the x-axis are the ones with the damaged DNA. And as you follow these men, you can see that the DNA damage has declined. It has gone down from 36.3 to 23.3. And levels above 21 are the ones which are the values which are bad. But if they continuously do yoga, the levels would come down to the normal levels. And you can see the second case in which the DNA fragmentation index was 34.3 and it has come down to 20.5. So yoga plays such a vital role in impacting the sperm genome. Now, as I told you that infertility is a psychosomatic disease, so we also studied the levels of the severity of depression in these individuals, and this was a randomized controlled trial. And we found that those people who practiced yoga actually showed a significant decline in the severity of depression, and this was because yoga actually resulted in an increase in the expression of various genes which promote neuroplasticity. For example, there was an increase in the brain-derived neurotrophic factor, the DHA, which is known as a brain vitamin, the levels of serotonin. A decline in the level of serotonin actually results in depression, and you can see that post-practice of yoga, the serotonin levels have gone down. And when people have depression and such high levels of stress, there is a disruption in the sleep-wake cycle. And we find that post-practice of yoga, there was an increase in the levels of melatonin. Now, this is a very, very important finding because not only does it regulate the circadian rhythm, but melatonin is a very potent antioxidant, and the highest levels of this are found in mitochondria. So it also helps to improve the mitochondrial integrity. In addition to that, we find that psychological stress also results in oxidative stress. The post-practice of yoga, there was a decline in the free radical levels, an increase in the antioxidant capacity, and a decline in the level of this mutagenic base, and also a maintenance in the telomere length. Now, it's been seen that people who have stress, anxiety, and depression, they tend to age faster. And we've now shown that if you practice yoga, you can slow down your rate of aging, and your biological age could be much lower than your chronological age. So we also see a decline in the cortisol levels, the stress hormone, an increase in the levels of sirtuins, and an improvement in the mitochondrial integrity. And what was also important was that we found that in people who have depression, they have lower levels of various tumor suppressor genes. I've just given you one example here. But post-practice of yoga, we actually found there was a significant increase in the expression levels of various tumor suppressor genes. I've given RB1 as an example, and genes which are helping to maintain the circadian rhythm. And there was an improvement in all the 4 domains of quality of life. So I would just like to summarize by telling you that yoga decreases oxidative stress and inflammation, helps to minimize the damage to the nuclear and the mitochondrial DNA, which helps to improve the genomic integrity, maintain the telomere length by decreasing oxidative stress and increasing the telomerase activity, helps to normalize the epigenetic marks, and thus yoga plays a very, very critical role in not only improving the testicular health, but also because it's a mind-body intervention, it helps to reduce the comorbid depression, and thus it should be practiced and used as an adjunct therapy in the management of male factor infertility. And thus it's such an important finding that since yoga targets the genome and the epigenome, all individuals who have any derailment of their reproductive health should be advocated to practice yoga. Thank you.
Thank you, Dr. Rima. That was truly an outstanding and very interesting talk. And it's very fascinating to see how the day-to-day oxidative stress can affect us at such a molecular level, cellular level, and even affect the genome of the sperm. So, and that is the very foundation of preservation of human species and building a healthier society. And it's truly interesting to see how it can affect the future generation in terms of not only the various genetic disorders or congenital disorders, but also the incidence of malignancies in the offsprings, as you say, and even the psychosomatic, various other psychological and disorders, etc. That can happen in the offsprings of people who are affected by oxidative stress.
Right, because majority of de novo germline mutations arise in the sperm DNA, not in the oocyte DNA. Majority of oocyte abnormalities are usually karyotypic abnormalities, but de novo germline mutations are usually from the sperm. So they are the cause of majority of the autosomal dominant genetic disorders and these complex psychiatric disorders, early pregnancy losses where there are paternal factors, congenital malformations, again, where there are paternal factors, all the female factors are ruled out, are all due to the problems in the sperm. And mainly, and these are all, the main thing is that these are all preventable because they are mainly due to unhealthy social habits.
Thank you, Dr. Rima. Very, very important insights. In fact, both the speakers, both the faculty have given very important presentations, and as audience, we should all learn from this and see how we can incorporate yoga in our day-to-day practice. And I myself feeling quite motivated today to see what best I can do to increase the exposure to yoga in my own life. Thank you so much. I think we are already behind time. There's no other question in the chat box, so we can close the session. Thank you, both the faculty. Thank you, audience, for being with us. And thank you, American Academy of Yoga and Meditation, for making this possible. This wonderful session. Thank you so much.
Thank you.
Thank you.
Thank you. Thank you very much, Dr. Dada and Dr. Deepak. And thank you, Dr. Saxena, for moderating this very, very interesting session. My take-home messages are 2 from here. One is that poor lifestyle choices can affect our sleep, decrease our resilience to stress, cause chronic illnesses, and even cause genetic changes and affect our offspring. And the yoga lifestyle, which includes not just pranayama and asanas and meditation, but also our thinking and eating habits, can actually mitigate most of these changes and even reverse many of them if practiced regularly. So that's a great message to take home, and explain so beautifully at the basic molecular level how yoga works to help us. So thank you all very much. And I also want to thank our tech team, Dr. Anjali Deshmukh and Sagar Shah. Thank you so much, everybody, for attending this. Our conference will resume again next Friday with a yoga session. So hope we have lots of attendees there as well. Thank you all very much. Namaste.